Okay, welcome everyone to the 51st episode of our webinar series of the ECOP program, Early Career Ocean Professionals program. This episode, Interfacing with the Ocean and ArtWaves Artist Residency project, will be moderated by Laura. So I'll give the floor to you.
Thank you, Anna. It's a pleasure. So, hello, everyone. My name is Laura Suaza. I'm from Colombia. I'm a geologist, and I am part of the ECOP program training and mentoring tax team. So, like Anna say, I will be moderating today's session. So please feel free to drop your name, profession, interest, LinkedIn. So the idea of this webinar of ECOP is to connect and strengthen this network in ocean science. So we can wait maybe a minute for more people entry.
Laura, do you want me already to share my screen?
Yes, I think you can start to share your screen.
Yeah,
Yeah, it's perfect.
Yeah. Okay.
Yes, maybe before we begin, just general indications. This session is being recorded, so it will be available on the ECOP YouTube. There is a Q&A box too, so you can write your answers anytime or in the chat too. Well, this is the episode 51 of the ECOP program webinar. Today's session offers a unique perspective to explore the intersection between science, art, and ocean literacy. So the idea is to, through creative approach and interdisciplinary collaborations, discover new ways to understanding marine biodiversity and communicating the complexity of our oceans to society. This webinar is titled Interfacing with the Ocean and ArtWaves Artist Resident Project. So we can welcome to Dr. Ruth Krause. She's a head of science strategy and management at Helmholtz-Institute for Functional Marine Biodiversity. Her work focuses on strengthening connection between researchers, institutions, and society, while supporting the development of young science and promoting the dissemination of scientist knowledge to broad audience. So thank you, Dr. Krause, for being here. It's an honor. Our second guest is Max Seeger. He is a freelance artist, designer, and PhD candidate. Max is a media artist whose work combines technology, interactive installations, and multimedia experience to explore relationship between humans and nature. So his work has received international recognition and have been exhibited across Europe and Asia. So we are very excited to learn more about ArtWaves Residence and hear about your experience and this connection between science and art. So welcome, Ruth and Max. Thank you for being here. So the floor is yours.
Thank you very much, Laura. Thank you very much for the invitation. Thank you, Anna. I'm happy to be here today and to introduce you to ArtWaves, an art science initiative that connects artists, scientists, and the wider public around ocean and marine biodiversity related topics. ArtWaves is a collaboration with the Helmholtz-Institute for Functional Marine Biodiversity, HIFMB in short, and the Hanse Institute for Advanced Study, in short HWK or HWK in Delmenhorst, also in the northwest part of Germany. And today I will briefly explain what ArtWaves is, how we work with artists and researchers together, and what we have learned from these collaborations. Let me start with 2 slides about us. about the institute, Helmholtz-Institute for Functional Marine Biodiversity. It's a cooperation institute between ARWI and University of Oldenburg. Maybe some of you know the ARWI, the major German research center focused mainly on polar research, and the University of Oldenburg, who's a relatively young university in Germany with roughly 15,000 students. So a rather mid-sized university. Also, it's in the state of Lower Saxony in the northwestern part of Germany. I'm sorry. We are young. The HIFMB is just founded in 2017. We have 90 in total, 90 employees, members, and guests. And last year we moved into our new building, which you can see here in the background, with 85 office workplaces and roughly 400 square meters lab space. Our mission is to understand the function of marine biodiversity. So from food webs, nutrient cycling to carbon storage and ecosystem stability. And we research how these processes support essential ecosystem services like fisheries, coastal protection, climate regulation, and so on. And together with social scientists at HIFMB, we also address critically the human dimension of ocean use and governance, which is fundamental to fully understand decision-making in marine conservation. But now let's talk about ArtWaves. ArtWaves is an artist in residence program, which we initiated in 2023. We had a one-year test run, which was successful, and then we planned to have it annually with annual calls for the entire duration of the UN Ocean Decade. We defined 4 objectives. The first objective is that we aim to provide insights into key aspects of marine biodiversity change. We want to promote public understanding and awareness of biodiversity change through art in a world that is affected by climate change. And with new formats, we aim to bring science into social and also political debates. And by creating new networks, we aim to increase the visibility of our research. To more effectively achieve these goals, we applied for the Ocean Decade Action endorsement, which we received in January 2024. The artist in residence scheme is such that we have individual artists each year for about 3 to 4 months. And the fellows live at our partner organization, the HWK, Institute for Advanced Study in Helmholtz, which provides an apartment and a studio space. And they work with us at the Institute. And here it is basically Stephanie Winner and myself, we ensure that the artist is well integrated into the Institute and its research. Now I have a short video that was prepared for the UN Ocean Decade Conference. It's just 40 seconds.
Kunst und Wissenschaft sind getrieben von Neugier, vom Explorieren, vom Verstehen, vom Ausdruck von Komplexem. Wir erleben, dass durch den kreativen Austausch Forschende neue Denk- und Arbeitsweisen mitnehmen und dass das Institut innovative Formate den Dialog an der Schnittstelle Wissenschaft, Gesellschaft und Politik vorantreiben kann. Darüber hinaus öffnet Kunst andere Wahrnehmungsräume und schafft neue Erfahrungen. Science is to recognize, art and design are to relate.
I forgot to mention that it's in German, but I hope you realize that there were English subtitles. Let me look back a little bit to our artist fellows that we had. We started the test run with Mita Mahatu. During her stay, Mita further developed her project on whale fall. Whale fall, you may know that, describes what happens when a dead whale sinks to the ocean, deep sea floor of the ocean, and slowly becomes a whole ecosystem itself, supporting many forms of marine life for decades. And her work consists of 9 pages of visual poetry that explored this phenomenon. And during her stay, she learned about environmental DNA research and sequencing methods, which study organisms in solution. Based on this, she experimented with letter forms and color coding. Here's another video. In doing so, she asked what voices and connections and possibilities for survival might emerge within this complex ecological and here symbolic mesh. Next was Anna Pasco. Anna has worked on art projects that explore symbiotic relationships, the ways in which we coexist with other species and with the environment. She refers to symbiosis with both organic and inorganic environments, combining art theory, pop culture, and science in her work. And she presents her practice in a variety of formats, demonstrating her skills in working with artificial intelligence, images, photography, ceramics, so on. And then last year, we had Antea Österreicher with us. She again had a completely different focus. She explored the deep connections between human and planktonic life. And these shared spaces, different ways of knowing, sensing, seeing, and breathing comes together and flow into one each other. And she asked how we come to know these invisible but planetarily important organisms through different practices, and how these interactions also transform or change us. This year we had Max, media artist from Berlin. He asked how we can make oceans more accessible to a wider audience beyond diagrams and tables. And in a couple of minutes, he will present his project interfacing with the ocean, so I won't tell too much here. But before I hand over to Max, where he can share more of his experience and lessons learned from his team, please let me spend a few words on what we, or better the scientists at HIFMB, learned from this artist in residence scheme. So we asked especially the early career researchers at our institute, and I thought for you ECOPs it might be interesting. What impact had the interaction on your work? And here, one shared key insight is the added value of having an artist within the scientific community. As one of us put it, science is not limited by a lack of creativity, but often by the repetition of the same methods or patterns of thinking. And art can help to open this space and to broaden how we think. Another quote was, working with different ways can take us out of our comfort zones and contribute to more diverse and resilient scientific practice. And several of us also reconsidered how we communicate our results, taking away practical ideas for presenting our work more clearly across disciplines and for communicating our main message to wider audiences. And I found following one very interesting. Collaboration also had lasting effects on how we perceive our own work. Many of us started to see our data differently and to recognize new value and even excitement in it. It helped me realize that my research can be meaningful and engaging beyond our immediate scientific context. And with these really beautiful and sometimes even touching experiences and impressions, I would like now to hand over to Max. I think, Anna, it's now time for you to put another question into the chat. Please feel free to answer this.
Yes, indeed. We had a quick poll question. I've just launched it, so everyone should receive a little pop-up screen with the question, in your perspective, what is the main value of having an artist in a scientific community?
Before I end, I would love to take this opportunity again to thank Max once again for his amazing, open, creative, and interactive work with us and for the time he spent at HIFMB. Thank you very much, everybody, for listening. And in case you have any more questions, please feel free to contact me anytime. Thank you.
I'll give it a few more seconds for the poll question, but thank you so much, Ruth, for this very clear and a nice introduction presentation. I'll end the poll question now. I see we have a few answers. So I see a response saying science communication, a way to bridge the gap between scientists and the general public and to generate impact. A second one is saying fresh new ideas and perspectives of how to translate scientific knowledge in a way that is understood by and engaging to a wider audience or general public. And a third one, sharing different points of views and different ways of thinking. And then a fourth one saying no answer, but maybe by the end of this webinar, there will be an answer. So thank you everyone for responding to this question. And I will close the poll question. And then Laura or Max, I'll leave the word to you.
Can you see my screen?
Yes.
Okay, perfect. At first, thank you, Anna and Laura, for the really nice introduction and for the kind words, Ruth. I'm really glad to be here today and talk a bit about my time at the HFMB in Oldenburg for this artist residency. At first, I want to just shortly introduce where I'm coming from. And then after that, show an older project that is also related to marine topics, just to give you an idea what I usually do. And then after that, I want to talk about more kind of insights and work in progress and results of the artist residency. So I'm Max. I've studied actually design at this beautiful building in Würzburg, which is kind of in the center of Germany. And after my studies in design, I kind of shifted a bit towards the, I would say, line between design and media art. And now, at the moment, I do live in Berlin and work as a freelance artist and designer. But I'm also pursuing a PhD in media art at the Art University in Linz. Where I investigate with like in a practical way how technologies can be used creatively in different ways to mediate the relationship between humans and nature. And this topic is also where my project at the HFMB fit in really well, and also some other things that I've done in the past, as you'll see. And yeah. I'm back in Berlin for a month now, but the 3 months before, I was living, as Ruth said, at the HWK in Delmenhorst, which was the hosting institution where I could live for the time of the residency, and I was mainly during this time I was mainly based at the HFMB in Oldenburg. If I'm not doing an artist residency like that, I'm working on all kinds of projects that are all roughly related to digital technologies, design and programming. But one topic that has always stuck with me is like throughout a lot of projects is on the one hand, exploring this connection between design and science communication or art and science. And on the other hand, somehow I ended up doing several projects on marine topics because I'm really interested in this personally. And that's why I want to show you one project I've done in the past, which is called Jewels of the Sea. And actually this whole project for me started way before I actually knew, because before this project, I was working on another work that where I was really fascinated about how universal patterns and shapes show up throughout nature in different kind of sizes and scales and through in different ecosystems and parts of the world. And during that time, I researched a lot on different shapes reoccurring in nature. And on some day on flea market, I stumbled across an old book from Ernst Haeckel, The Art Forms in Nature, which was published somewhere in, I think, 1900, like early 20th century, late 19th century. And I've seen his drawings and I was kind of confused because I thought I've researched all these organic shapes and all these kind of nested, irregular, unsymmetric shapes. And then I was looking at that page and I thought this must somehow be some kind of artistic exaggeration. But then, like 2 minutes later, I was Googling diatoms, and as most of you probably know, and as I didn't before, diatoms really look like that. So I was kind of fascinated how these symmetric forms show up in this kind of variety, but also symmetric perfection, I would say. So I was kind of hooked, and I kept researching on diatoms. And what really struck me as well is in how diatoms have been a topic of artistic and science collaboration or joint interests for a really long time, actually, especially in the 19th century, early 20th century. For example, this art practice of laying diatom images was really famous during the Victorian age. Uh, Art practitioners would just take sea samples and then under a microscope with a really specialized glue that's drying really slowly, they would use like pig hair, like from small pigs, to under the microscope pick the specimens that they want and assemble symmetric small images from the diatoms. And if you look at it, how tiny they are, I think they're some of the tiniest beautiful images I know. Um, and there's also there are also these drawings and stories about how in Victorian age in like saloons, people just met with these specialized microscopes to look at these small arrangements of diatoms, but also sometimes butterfly scales, which is kind of like how we would meet today to watch a movie, but they looked at microscopic samples, which is kind of interesting, I think. And also during that time, a lot of architects and artists have been inspired by the shapes, not only of diatoms, but in this case, rather of radiolaria. This is from the gate from the Paris World Exhibition, 1900, for example. Or Richard Buckminster Fuller, for example, a famous British architect, he was talking a lot about the shapes of diatoms and radiolaria and how they inspired his style of building. Or also Frei Otto, the German architect who built, for example, the Olympic Stadium and Park in Munich. He even had a research group in Stuttgart researching diatom shapes for lightweight construction for, I think, 10 years. So yeah, I was kind of really interested in the history of diatoms for a while and how it has influenced also art and architecture. But then came back and thought what was the thing that I initially surprised me most? And this was this kind of symmetry um, that you rarely find anywhere else in nature. So I was asking myself the question, where does it come from? But of course, quickly realized I can't answer that at all. But the next question I had was because I'm working a lot with simulation and computational techniques, is can I kind of replicate algorithmically how this symmetry and also variety in shapes comes into place? Um, and of course, I did some research, and of course, I wasn't like exactly the first one asking that question. And some of the things I stumbled upon was, for example, the super formula by Belgian biomathematician Johan Gillis. He proposed this one formula, I think like 15 years ago, which with just changes of parameters covered a huge variety of like natural shapes or shapes reoccurring throughout nature. And also in some of his early papers, he just besides petals and snake houses, for example, he also that's him. He also mentioned that diatoms could be a use case where this formula could come in um, to simulate the shapes. And he's actually a really nice guy that I when later met in Brussels. But before meeting him, I actually kind of took his formula and tried to play around with it. And if you look at this chart of different shapes that you can achieve with different parameter combinations, maybe you can already see that some of them kind of look like a 2D version of diatoms. So what I did in a kind of longer process was to, at first, what you see on the happening on the right was generalizing his formula from 2D to 3D geometry, and also using the symmetry axis that he uses for calculation in the formula to extrude this kind of stria that would run through some of the skeletons of some diatom species and also the small thorns, the spicae. And on the other hand, I was interested on this really in this really special surface structure that diatoms have, because when researching this, I found out that some people have dealt with the like overall geometric forms of diatoms, how to simulate them. Some others with the how to simulate the surface structures, but I haven't really found anything where somebody had tried to bring these two things together. So that was kind of the point where I thought it would become interesting if I tried to do that. So I used, for example, one algorithm for the smallest packing of spheres and circles, which was also proposed by this research group by the architect Frei Otto to simulate the surface structure on diatoms. You can see that happening in the center. And another paper proposed to use an algorithm that's using the Fibonacci numbers to also, to also calculate the arrangement of pores on the surface. And this took actually quite a lot of back and forth and time and trial and error to come up at the end with an algorithm that had kind of 50 input parameters which you could tweak. But at this point, I knew that I was coming somehow close to what could look like some diatoms, but I was totally missing the like the background knowledge about different taxa, or like on a more species level, what would be the taxonomic features that I could match with my algorithm that I was working on. So I reached out and like on to several people on forums on the internet, and I somehow found Matthias Burba, who was actually working as a taxonomist on the site for Max Planck Institute. He was usually trained as a forensic scientist, but he has a really well knowledge on taxonomy for diatoms and foraminifera. And he actually took the time and to collaborate with me, and we started this process of going back and forth. Now we're looking kind of at the final algorithm, tweaking the algorithm parameters towards a certain kind of, I would say, more on the genus or family level diatom. And he was feedbacking me to tell me this has to look a bit different. This should look more like this. And so we kind of went back and forth to dial in, I would say, around 100 different shapes that should match certain diatoms on a genus level. And I was also working on the look of it. which kind of has this now opal iridescent look, which resembles a bit the refractive and iridescent abilities that diatoms, because of their glassy houses, as you see colloquially, have. And also this whole algorithm was running in real time. So you could really in real time tweak the parameters and see the shape moving. Because of the way it was programmed, there was also it was a benefit that you could also easily simulate these kind of colonies that especially centric diatoms build, which you can see here. And yeah, after refining this process, I had in the back of my mind this kind of arrangements of diatoms, which I've seen. So I tried to kind of match some of them, or just see if I lay out like some of these digitally generated diatoms, would they match? How closely would they match? Because of course, they don't match, because it's computer simulation. You always get like, it's always an approximation, but how close is the approximation? And I would say these shouldn't like resemble the same species or anything, but just like to give a general overview. At this point, I was kind of confident and happy with the result. That I got kind of close, of course, with the help of the collaboration also with Matthias, to match some of the features of actual diatoms with these digital models. Here's, for example, like comparison of that should be like a Discus genus level diatom. And if you compare these two, you at least see, I mean, the real one already is a bit broken, but you at least see how close I got. It's not like perfect in every tiny detail, but the overall ability, the overall appearance is there. And after having this algorithm, I thought about how to make this also, how to show this to a wider public, and also kind of convey I would say, curiosity and joy diatoms have sparked for me. And during that time, I also I bought a microscope, and I looked at diatoms a lot under the microscope. So I wanted to make this kind of part of an artistic installation. So I built this tiny DIY camera on top. So you could just, with the camera, look through the microscope. And I also reached out to somebody else during that time, which was Dr. Michael Kloster from the University Duisburg-Essen, because he was actually working on automatically recognizing different species of diatoms with artificial intelligence. Still back then, I think this was 2021. And he actually helped me a lot to kind of train my own small image recognition model for diatoms, which wasn't working, of course, as sufficient as his own, because I didn't have like the fancy, super expensive microscope. And also I didn't have like the time and expertise he had, but by taking a lot of diatom images with my microscope and training a machine learning model, we could at least on this higher genus level recognize diatoms, which you can see happening here. And the installation that I exhibited at the end was kind of, we can look at it together. It's a table where you could just pipette water samples and then lay them under the microscope and look at them. You could use all the mechanics of the microscope to zoom and to just go back and forth with the sample. And whenever you find a diatom, it was automatically recognized by the machine learning model. And then kind of genus or family level diatom was automatically generated by this algorithm I showed you earlier. So you kind of got a digital model for the actual diatoms you've discovered. And because the model was digital and was 3D, you could also use hand tracking to actually go in with your hand and grab the diatom and turn it around, which is kind of giving you was using technology for this connection to something in the microcosm that you usually don't have. You usually don't have the ability to like rotate on the microscopic level. But with these digital models, you at least have kind of the feeling of that. Yeah, that was pretty nice because the project got some good got really interesting feedback on the project, and I had abilities to exhibit it on in kind of artistic context, which was really good for me. But the thing that really surprised me was that, like, I don't know, 3, 4 weeks after publishing the project, somehow I got mails from people that I didn't know, and these were actually not coming from the artist community, but rather from the science community, which was really interesting for me happening. So, for example, somebody reached out and he wanted this 3D models of the diatoms because they wanted to train like a low-cost microscope with these diatom models, and they didn't like back then. It was pretty complicated to model so much or do like microscopic photogrammetry on so much different species. So they wanted them kind of a baseline for training. Yeah, somebody else reached out and wanted the algorithm to use in his own research, or like a lot of people actually requesting these 3D models back then, which was kind of a thing that I didn't see coming when I started working on this. Yeah, I'm just like dropping something in here. And also some people so for example, this mathematician Johann Gielis reached out because he really liked the models for his own papers and was really happy to see that the thing that kind of he in his head already knew, that you could actually simulate the shapes of, for example, diatoms with this algorithm, that somebody from a different perspective also not only calculated it on a mathematical level, but also kind of came in and gave it a visual, like an aesthetic quality that he could use for his work again to show what his algorithm actually does. So just skipping through these, but I was really surprised that kind of this feedback came from a place I didn't actually expect. And I was even more surprised that people were approaching me with ideas that I didn't have when working on the project. And this was something that really, again, convinced me that there's something in this art science collaborations that when like bringing together perspectives like this, that for both sides there can happen a lot of unexpected things and ideas. And if anything about this project, really, if I hope for anything for this project, it was that thinking of all the old time when artists and scientists worked together on diatoms like 100 years ago. Maybe for myself, this project brought this back a little bit, this feeling of working together and bridging perspectives. And this is one of the reasons why I keep why I'm really, really into these kind of collaborations and try to approach scientists, researchers, or institutes. For example, like the HIFB for the ArtWaves Residency, to work together on projects like this and having the ability to, without like prior knowledge of what you'll actually do, because you should have the time to develop a common ground together and ideas together, work together on different kind of topics. And while staying at the HFMB, I really had the opportunity to do this for this artist residency. I would say almost the first whole month I could just sit on my desk in the institute, join every lab meeting there was, talk to people. Everybody was really open to show their research, just brainstorm ideas how this could be translated in different ways. And after I can't really show you all this trajectory, but like after kind of 1 month of being at the HFMB, I settled with Dorothy Hodap, who's Dr. Dorothy Hodap, who's working at the HFMB to have a closer look at one of the data sets she has assembled. And I don't want to make you dizzy for too long, but it was like she assembled a really, 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 really big data set of biodiversity data from the German North Sea coast, like along the coastline, biodiversity on like fish, phytoplankton, zooplankton, benthos, plants, and birds. And these were like, I think, around 360,000 different samples and over 2,100 different species showing up in the samples. And we were thinking about how it would be possible to make this approachable for a more wider audience. The first problem was we needed to make it also approachable for me. So I saw it while I had the feeling only looking at tables didn't really get me closer to what's really in the data set. I started to build a small like data visualization website for myself to really only for myself get what is inside the data set. And you could, for example, click on every site where there was samples in this data set and then click on every species that was in the data set. And something that I really that I recognize really fast is because I don't have a connection to all the with having no background in biology, I don't have a connection to all the scientific names of organisms. But just like scraping images of these species online already gave me a kind of feeling of what's actually in this data set. So this was kind of the first thing that struck me, that giving these names an image or something that's kind of relatable is making a first connection for me to really think about all these kinds of different species with all their different features and traits, and also their different occurrence or abundance in this kind of area, and makes it more approachable for me. But still, I was feeling like I I kind of had an idea what I was looking now from like a numbers perspective, but I didn't really know what it's like to generate these kind of data sets. So I reached out to several people, and also a lot of people from the HFNB connected me to colleagues of theirs, and I was able to join like some field trips, for example, to Spiekeroog, where they did where I could join the Dynady project, where they drilled for pore water and were sampling bacteria. And it was really interesting for me to see how this differs. I'm telling no new thing to a lot of you guys probably, but how different fieldwork feels from sitting at your desk and analyzing these kind of data. And apart from this field trip, like the most interesting thing for me was probably that I could join a research cruise on the North Sea, actually sampling benthos in a similar way than also some of the samples that were taken inside the data set I was working on. And I could join this team for the cruise. And it was a really interesting experience for me, not being on the sea longer than a few hours before. And also what was really productive for me for the later project, I think, was seeing how this is like how these data sets are not coming from nothing, how people are actually being out there generating them, and how they actually what I wanted to show is probably coming soon, how they actually approach also these animals, and however tiny and special they are, with a great curiosity and care. And this is something that I thought that when communicating to a more broader audience what is about, like what is research on biodiversity about, usually this is kind of on the way from like field work to data analysis to communication. This is something that's lacking or getting lost on the way, that maybe is really can be really present during the process. So I wanted to kind of bring this back, the curiosity, also the visual connection you have with an animal in front of you, and also the touch kind of as the physical action as a gesture of also care and curiosity. And coming back to my small web experiment I made, what I did then was using all these scraped images of the species to use a workflow with a neural network that can generate 3D models to try to actually generate 3D models of the species that were in this data set I was working with. And this work, after a lot of back and forth, this kind of worked well. Not for like every species, but because for some of the species you don't even find images yet, for some planktonic species, for example. But for the ones where I could find proper images, you could generate 3D models of the species that were actually in the data set. And I wanted to use these to make them kind of to give like a wider audience an idea of what is living in the North Sea and how does the abundance of these species change actually. And for this, I also disassembled on a really old like computer screen, because when disassembling it, you can kind of make these transparent screens out of it. And I used this for the installation. That was kind of the result of the residency. So you can see here, there was a tiny aquarium, and over this, there was this transparent screen that you could look through. And in the exhibition, this was set up in a way that you had actually water inside the aquarium. So you could, if you wanted, stay above water, but you could also put your hand inside the water. So you're actually making yourself wet and getting into the water. And there was a camera sitting underneath the aquarium that was tracking your hand. And with your hand, you could navigate through the data set in different ways. For example, at the beginning, you could, on the left, you could use your hand to select certain samples and data points along the coastlines that you wanted to look at. And then with your other hand, you could jump into the water, metaphorically, and with like one gesture, zoom in. So go from the macroscopic level of seeing fish into really the microscopic level of seeing the plankton that is in the data set. And also you could, with another gesture, go from the upper levels of the water column down to the seafloor, where you would find the benthos, for example. And for me, it was... Oh. Sorry. I hope you see that again. Somehow everything just collapsed. Okay, I have to share again. Just a few slides left. And for me, it was really interesting when we exhibited this. It's currently in the state of a prototype. I would say that I'm still working on, because I want to use this for my PhD project, but still as a prototype, exhibiting it to people from the field, from the institute, but also people that have nothing to do with marine biology was a really interesting experience. For example, showing this to somebody that actually doesn't know a lot about the topic, and having a researcher standing next to me explaining, oh, this abundance curve actually makes sense because there were small abundance curves showing up when you touched species. That was kind of cool, and really showed to me that they're fusing like the skill set and the abilities that, for example, in this moment we both had really really resulted in some kind of surplus or value for communicating what is actually happening in our oceans, like in a tiny way, I would say. But for me, it was really, really interesting experience. And I would like to end with that and like to say thank you again to the HFMB and the HWK for giving this opportunity to me. And for myself, only say that I'm always super interested in these kind of art science collaborations, and I'm really happy to hear questions, but also really happy for everybody who wants to reach out later on and maybe discuss ideas or just say hi. Yeah.
Thank you, Max, and thank you, Ruth, for this opportunity. I love having the opportunity to moderate this webinar. It reflects that values that I really believe in. In my university, I work in a project with art and science too. So I love these ways to make it science accessible and building bridge between knowledge and society. So thank you, Max, for this project. I take a class about microfossils too, and I have to say that they are amazing. Like you say, the symmetry and the ways that they create different geometry forms are amazing. So well, it inspired to me to make this project like more real, connect science with community. For example, here in Colombia, I work with rural communities, like making art in these rural places with the rural schools. And we include painting, circus artists, like drawing during the field trips. So it's really interesting to like mix these activities with technology too. So I'm very happy to receive this information and to know that there are many opportunities to work too. So it's a pleasure to moderate this webinar. If someone have questions or comments or anything you want to know, be free to ask.
Yes, and maybe a small note on that, that if anyone has a question, you can put it in the chat or the Q&A box. But if you raise your hand, then I can also enable your microphone and you can have a conversation with Max and Ruth. So whatever is most comfortable, if you have any questions or you want to share something. And in the meantime, I will share another quick poll. Let's go back. You should all see a little screen popping up with the first question. Do you have experience with a science-art collaboration? A second question, if you have experience with a science-art collaboration, how did it go? And then a third one, are you interested in exploring science-art collaborations more? And maybe in the meantime, Max, I have a question for you. Since you are, I don't know too much about technology and neural networks, but since you are modeling these tiny organisms, do you think the next step would be to model an ecosystem and see like maybe if food resources disappear, what happens, to visualize those things in 3D?
Yeah, that would be super interesting actually, and it's something I also discussed with researchers at HFMB, because actually there's a group that's interested in exactly these kind of things. But I think what you always have to keep in mind is that this is super complex in a way that even the people that are like, I would say, most sufficient in modeling these kind of systems, they either choose to model it on a meta scale and then missing out like a lot on the details, or they're modeling like really tiny details and missing out on the bigger picture. At least this was kind of what the modeling people at HIFMB told me. So it's a really difficult topic because then like when you really want to model it sufficiently, you're moving into really abstract space. And bringing this back to the less abstract space is a big challenge, but definitely one I'm interested in.
Mm-hmm. Okay, so more collaborations between the people that work on a tiny scale and the meta scale.
Yeah, but yeah, that would be interesting. But I think the thing I learned again also at this residency was that things are like you can never grasp the entirety of like uh like ecologic systems. It's just really difficult, like with computational resources, we're far away from that. So you always have to set a focus on like one thing. And but then I think it can be really powerful to show this.
Mm-hmm. So I see that we have some answers for the poll that we send out, so I will end it and then share the results. But we have to go to view details. So do you have experience with a science art collaboration? More than half, well, 2 people say yes, 1 person says no. If you have experience with a science art collaboration, how did it go? And then there's a few words coming up. Collaboration, chemist, physicist, material project, collaborating, research. And then 1 person is saying, I was collaborating with chemists and physicists on my material research project. The collaboration was very enriching to the project. And then for the question, are you interested in exploring science art collaborations more? Someone is saying yes, as someone with a background in both marine science and an art, it's a topic I'm interested in exploring. And then someone else is saying yes, absolutely, in the future with marine scientists. So I will stop sharing these results.
Well, I have another question. Maybe if, for example, we are interested in participating in the program, like how we can like entry our, which are the dates, or how we can be like more, I don't know, informed about the program.
I can maybe answer this. As I said, we run this call each year. We will open it again in summer or after the summer break, and then we expect the next artist to start beginning of next year, so 2027. And there's more information on our website, which I copy into the chat right now, and you can also always contact us and read on our website, follow us on Insta or any other channels, or just write us an email. That's all fine.
Okay, thank you.
Okay, if no other questions are coming in, then maybe we can end here.
Yeah, so thank you, Max, and thank you, Ruth, for being here. Thank you, Max, for expose your project. It's really amazing. I hope that more scientists like try these new ways to to expose the science, so and to connect with artists. So thank you, and thank you, Ruth, to support science and art too. So will it will be like amazing to be in the project in the future. So thank you for the information.
Thank you both so much.
And thank you for the opportunity. Thank you very much.
Yeah, thank you for having us.
Thank you, and hope to see more of your work and for the following participants of the project, of course. Okay,
Yes,
and thank
yes.
you everyone for being here in this webinar. and have a lovely day, evening, morning, wherever you are.
Thank you, Anna. Bye.
Thank you.
See you.
Bye.
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 Bye.